Hey there, fellow scientists. Welcome. You know better than anyone how long it takes to bring a new drug to market. We're talking years and literally billions of dollars. Well, in this explainer, we're going to dive into how artificial intelligence is way more than just a buzzword. It's actually becoming a core tool in the lab, and we're going to show you how you can get in on the action.
So let's start with this question, because this really gets to the heart of the massive bottleneck in modern medicine. Think about it. Over 90% of drugs that go into clinical trials just fail. For decades, the process has been incredibly slow and expensive. But the answer isn't just about doing the old things faster. It's about a whole new way of thinking, one where we can finally use all this data we're generating to let machine learning find the insights for us.
So, how do we actually break that bottleneck? Well, on the left, that's the path we all know, right? It's that long, winding road of painstaking trial and error. But check out the right side. That's the new paradigm. Instead of walking, we're basically using machine learning to launch a rocket straight to the target. It helps us rapidly screen and prioritize molecules, which just dramatically boosts our chances of success from the very beginning.
And that's the key thing to realize here. This isn't just about having faster tools. It's about a completely new way to ask scientific questions. It's a true paradigm shift. We're moving from testing our hypotheses one by one to discovering these incredible patterns in massive data sets that we as humans just couldn't see before.
So, what even is machine learning? To put it simply, it's all about teaching computers to learn from data so they can make predictions. And the magic word there is learn. We are not writing a specific rule for every single possibility. The machine figures out the rules all by itself.
And this analogy is just perfect for getting your head around it. You wouldn't hand a kid a 50-page flowchart on how to identify a cancerous cell, right? No way. You'd show them thousands of pictures, some healthy, some cancerous, and eventually they just get it. They start to see the patterns intuitively. That is exactly how machine learning works.
Which brings us to this core difference. With the programming we're all used to, you give the computer data and the rules, and it spits out the answers. But with machine learning, you flip it. You give the computer the data and the answers, like this image has cancer, this one doesn't. And the machine works backward to figure out the rules for itself. It builds its own model that actually gets smarter the more data you feed it.
Okay, so now that we've got the basic idea down, let's peek inside the AI toolkit. There are a few key methods that really drive this whole thing forward.
You've basically got three main flavors here. There's supervised learning, which is what we did with our cancer cell example. You need neatly labeled data. Then you have unsupervised learning, which is all about discovery. You just throw your data at it and it finds hidden patterns you didn't even know were there, like new ways to cluster patients based on their genetics. And finally, reinforcement learning, which is all about trial and error, like an AI agent testing millions of molecular combinations to find the one that works.
And then there's the big one you've probably heard of, deep learning. This is a super powerful subset of machine learning that uses something called neural networks. They're these really complex structures that are actually inspired by our own brains, and they are incredible at learning highly complex patterns from images, text, or biological data.
Okay, so that's the theory. Cool stuff, right? But what does this actually look like out in the wild, in a real lab? Let's check out the breakthroughs that are happening right now.
I mean, the impact is already massive, and it's touching pretty much every part of the research pipeline. We're talking about accelerating that initial hunt for new drugs, automating the painstaking analysis of medical images, decoding the very language of our genes and proteins. It's all paving the way for truly personalized precision medicine.
And if you need any more proof that this is for real, look no further than this. In 2024, Nobel Prizes were given for the foundational work in neural networks and for using AI to predict protein structures. For us scientists in the lab, this is absolutely huge. It's a massive validation that AI is a core tool of discovery, not just some niche curiosity.
So let's dig into that protein world for a second, because this is so cool. One of the best ways to think about proteins is to see them as a language. They're built from those 20 amino acid letters. By analyzing millions and millions of protein sentences, machine learning can actually learn the grammar, the rules that determine how a protein folds and what it does.
And here is a stunning, real-world application of that idea. To predict how SARS-CoV-2 might evolve, models were built to ask three simple but critical questions. 1. Is a potential mutation even accessible to our antibodies? 2. Does it change the virus's shape enough to hide from them? And 3, and this is crucial, does the mutation actually hurt the virus's ability to survive and replicate? This is how we can start to design future-proof vaccines.
Okay, but of course, with this much power, you've got to be smart about how you use it. This is not some magic wand, and as a scientist, you need to understand the challenges and, the practical rules of the road.
It really all boils down to this one simple, timeless principle. An AI model is only as good, as accurate, and as unbiased as the data you feed it. Your data quality is everything, period.
And two of the biggest traps you can fall into are called overfitting and underfitting. Overfitting is kind of like a student who just memorizes the answers for one specific test. The model gets really good at your training data, but it fails completely on any new data. And underfitting is the opposite. The model is just too simple, and it misses the actual biological signal you were looking for in the first place.
And as we start using these tools more and more, there are some really important practical questions we all have to ask. Like, can we actually explain why a model made a certain prediction, or is it just a black box? Is our data following the FAIR principles so the rest of the community can use it? Are we protecting patient privacy? And who's accountable when an AI gets it wrong? Thinking about this stuff up front isn't just bureaucracy, it's just good science.
All right, so we've seen the potential, we've looked at the challenges, now let's talk about you. This is a new frontier and it is wide open for you to explore.
And I'm guessing the question on your mind right now is probably, okay, this is amazing, but where on earth do I even begin? Well, the good news is, getting started is way more accessible than you might think.
First off, just start learning the core concepts. You really don't need to be a programmer to grasp the principles. Then, collaborate. Find the data scientists and the computational folks at your institution. But here's the most important thing. Never, ever forget that your deep biological knowledge, your domain expertise, is the most valuable piece of this puzzle. You're the one who knows the right questions to ask.
And that's what I want to leave you with. We've been talking a lot about predicting what already exists in nature, but what if the real future, with this technology in your hands, isn't just predicting nature's proteins, but designing brand new ones from scratch? Entirely new medicines, new biological systems. The next huge breakthrough is out there, just waiting.